morning in Taiwan and good evening in St. Louis. I'm Dr. Chaur-Jong Hu, the Director of Dementia Center, Shuang Hospital, Taipei Medical University. The Dementia Center is funded by National Health Research Institute this year to promote dementia care and update dementia knowledge for physicians and dementia care team members. We will have 11 masters contributing to this continuous education program, Frontiers in Dementia Research Master Lecture. The master of this month is Professor John Morris from Washington University, St. Louis, US. Professor Morris right now is Harvey A. and Dorismae Friedman Distinguished Professor of Neurology, Director of Ninth Alzheimer's Disease Research Center of Washington University. He is a global leader of Alzheimer's disease research. His research focuses on improving the diagnosis of early stage of Alzheimer's disease, clinical pathological correlation in dementing disorders, evaluating new drug for treatment of dementia, establishing phenotypes of inherited form of Alzheimer's disease and other dementias, and identifying preclinical stage of Alzheimer's disease by biomarkers and neuroimaging. I have ever studied in Stroke Center of WashU in 2000. I usually attend the research meeting and learn a lot about dementia knowledge in ADRC led by Professor Morris. The weather of St. Louis is terrible right now, but Professor Morris still will answer few questions after his video presentation. He also donates his honorarium to us for helping dementia research in Taiwan. I want to express our great thanks to him. The title of his talk today is Prevention Trials in Alzheimer's Disease, the Role of Preclinical AD. Let's welcome Professor Morris. Okay.
Thank you very much. Pleasure to be here.
Hey.
I'm John Morris, the director of the Knight Alzheimer's Disease Research Center at Washington University in St. Louis, Missouri, the United States. And I'm very honored to be asked to provide this keynote address as part of the Master Lectures of Frontiers in Dementia Research, sponsored by the Dementia Center at Shuang Hospital of Taipei Medical University. I've been asked to speak on prevention trials in Alzheimer's disease, the role of preclinical Alzheimer's disease. My research is supported from grants awarded from the National Institute on Aging of the National Institutes of Health in the United States. I have some consulting relationships via my participation in advisory boards to a number of research programs and foundations. I have no connection with industry or pharma-sponsored trials. I first would like to thank all of the faculty and staff and advisors for the Knight Alzheimer's Disease Research Center. At the top of this composite, I am in the center as the director, but I have three associate directors, Randy Bateman, David Holtzman, and Carlos Cruchaga, and also our executive director, Krista Mulder. The therapeutic landscape for Alzheimer's disease began with an approved drug in 1993 in the United States, Tacrin, trade name Cognex, a cholinesterase inhibitor. And shortly after that approval, 3 additional cholinesterase inhibitors were approved, donepezil, rivastigmine, and galantamine, followed by an NMDA receptor antagonist memantine. All of these drugs are approved for the symptomatic treatment of Alzheimer's disease. However, there has been 2 decades of trials of putative disease-modifying therapies. And in 2021, the first such drug, aducanumab, was approved by the Food and Drug Administration in the United States. Aducanumab is a monoclonal antibody directed against an amyloid beta epitope. The antibodies were discovered by looking at older individuals who remain cognitively normal and assessing their anti-A beta antibodies with the assumption that this provides their resilience against developing Alzheimer's disease. Aducanumab binds aggregated A beta, not the monomer. The study has been sponsored of aducanumab has been sponsored by Biogen with support from ASI. And there were 2 identical phase 3 studies of intravenous aducanumab. And the studies were halted for fear of futility, but reanalysis of the data suggested that there could be a potential benefit of the drug. However, this potential benefit was only suggested by secondary analyses. Both trials failed to meet the pre-specified endpoint for efficacy. Nonetheless, the company submitted the data to the Food and Drug Administration that, against the advisory committee convened by the Food and Drug Administration that recommended it not be approved, the FDA did approve aducanumab on an accelerated approval mechanism in June 2021, based upon the drug's ability to reduce amyloid plaque burden. That is, they did not approve it because of demonstrated clinical benefit. There is no clear clinical benefit based upon the halted trials, but on this surrogate endpoint of reducing amyloid plaque burden. Upon approval, Biogen said it will charge $58,000 in US dollars for 1-year treatment with aducanumab. This was a very controversial decision, very controversial price, much scrutiny in the United States about this. And in fact, the Food and Drug Administrator's acting commissioner, 1 month after the approval of the drug, asked for an investigation to see whether the part of the FDA that approved the drug had an improper relationship with Biogen and whether any rules have been broken. That investigation is still ongoing. Because the drug is so expensive, everyone in the United States has been waiting to determine whether the insurers will reimburse for the cost of the drug. In the meantime, in December 2021, the European Medicines Agency declined to approve aducanumab. Biogen reduced its price almost in half in December of 2021 because very few people with Alzheimer's disease dementia were willing to pay the $58,000 out of cost. Just last month, January 2022, the major reimbursement agency for individuals in the United States 65 and older, the Centers for Medicare and Medicaid Services, agreed to reimburse the cost of aducanumab treatment only for individuals who were enrolled in approved clinical trials. That is, it is not available for clinical practice and the reimbursement of those costs. The decision of the Centers for Medicare and Medicaid Services seemed to have this approval, limited approval, covering all potential anti-A beta therapies, including those for which we are to learn the results from pivotal studies in the next year or two. Gantenerumab from Roche, lecanemab from Eisai, and donanemab from Lilly. It's difficult to know from the phase 2 studies of aducanumab whether any of the findings will pertain to the large majority of persons with Alzheimer's disease, because the participants in the trial did not reflect the diversity of the United States population. Only 19 of over 3,000 individuals in the trial were Black, for example, less than 1%, whereas the US population is 13% Black. And participants with common comorbid disorder, diabetes, and so forth, were ineligible to participate. And only participants with very mild symptomatic Alzheimer's disease were enrolled in the trial, whereas most individuals with Alzheimer's dementia have more advanced symptomatic disease. Aducanumab is associated with adverse effects, principally amyloid-related imaging abnormalities, mostly edema, occasionally hemorrhage. These abnormalities occurred in 35% of the treated patients in the 2 trials. 25% of the patients were symptomatic, and a few developed serious concerns, including seizures and microbleeds. The approval of aducanumab highlighted that in the United States, our clinical practice infrastructure is really unprepared should aducanumab be administered on a large scale. We have inadequate diagnostic capacity to identify people with very mild symptomatic Alzheimer's disease. It's uncertain how individuals will be shown to have the target, that is cerebral amyloid plaque burden, because the cost of amyloid PET are not reimbursed. Cerebral spinal fluid determination is not highly accessible. There is hope that perhaps plasma-based measures of amyloid will be useful in this regard to document that an individual with very mild clinical diagnosis, very mild symptomatic Alzheimer's disease, indeed has the likelihood of amyloid plaques. There are limited infusion centers in the United States to handle a large number of aducanumab infusions. And because of the potential side effects, amyloid-related imaging abnormalities, serial MRI scans will be needed to detect potential side effects. This has been, aducanumab comes at the end of 2 decades of looking at putative disease-modifying therapies for Alzheimer's disease. Since 2001, all have failed to demonstrate clear clinical benefit. This failure has been interpreted by some to challenge the amyloid cascade hypothesis of Alzheimer's disease. Here is a partial list of anti-A beta therapies that have been initiated since 2001, targeting amyloid either through immunotherapy or through inhibition of secretase, both gamma secretase and beta secretase. All of these studies have failed to demonstrate clear, clinically meaningful benefit. I put a question mark next to aducanumab at the bottom because the company believes there is benefit, but not with their pre-specified measure of efficacy. However, it's not just the failure of amyloid therapies in symptomatic Alzheimer's disease, but other therapies targeting non-A beta mechanisms have failed to show benefit also. These non-A beta mechanisms are not as thoroughly studied, but looking at cholesterol, inflammation, and insulin receptors, and mitochondrial function, and so forth, show no benefit either. So it's not simply amyloid that fails. To look at the amyloid cascade hypothesis of Alzheimer's disease, we recognize that Alzheimer's disease has a hallmark progressive cerebral A beta deposition. We know that rare pathogenic variants are causative for early onset Alzheimer's dementia, and they all in common have an overproduction of the highly aggregated form of amyloid A beta 42. And if one inherits one of these causative mutations, the likelihood of developing early onset Alzheimer's disease is nearly 100%. Additionally, individuals with Down syndrome have trisomy 21, it's chromosome 21, where amyloid precursor protein gene is located, overproduce A beta throughout life, and almost all Down syndrome individuals have neuropathologic Alzheimer's disease, also very early onset. A rare missense mutation in the amyloid precursor gene results in decreased A beta production, and those individuals are unlikely to develop Alzheimer's disease dementia. So genetic studies point clearly to overproduction of A beta 42 as a mechanism causing Alzheimer's disease dementia, and decreasing A beta production protects against Alzheimer's disease. So the amyloid cascade hypothesis can be rewritten to indicate that a disruption of A beta homeostasis, in this case overproduction, is the initiating event in Alzheimer's disease. Once amyloid beta is deposited in the cerebral cortex in the form of plaques, an initiation of a complex pathophysiologic cascade occurs, including tauopathy, neuroinflammation, other mechanisms that in turn result in neurodegeneration and cause the symptomatic expression of Alzheimer's disease. So amyloid plaque deposition is necessary but insufficient to cause the Alzheimer's disease dementia syndrome. One of the anti-A beta monoclonal antibodies, gantenerumab, clearly shows that the drug engages its target, amyloid plaques, and reduces the amount of amyloid burden in the brain. The top row, baseline, shows the amyloid PET scan of 5 individuals prior to treatment with gantenerumab. At week 52, 1 year after gantenerumab treatment, you can see in the second row for each of those 5 individuals, a lowering of the amyloid burden is demonstrated by amyloid PET. So why are these drug therapies failing to show clinical benefit? Perhaps the drugs are ineffective, but it seems that they do engage with amyloid plaques and are effective in reducing amyloid beta burden. Maybe the doses are insufficiently high. For early studies, there was no clear eligibility criteria requiring demonstration of amyloid burden with amyloid PET scan. Simply people with a clinical diagnosis of symptomatic Alzheimer's disease were enrolled. Some of those trials, it was later found, enrolled individuals up to 1/3 of the total cohort who did not have Alzheimer's disease. And it still is possible that the targets, such as A beta, may be incorrect. But another reason for the failure of disease-modifying therapies is that they are initiated too late in the disease process. Some studies at Washington University several decades ago, looking at serologic counting of neurons in medial temporal lobe structures, found that in individuals who died, who were cognitively normal and had very few amyloid plaques and very few tangles except in entorhinal cortex and in the hippocampus, had no cell loss with age. They were clinically normal at time of death, and their pathologic diagnosis was normal as well. In contrast, individuals who died in the very earliest symptomatic stage of Alzheimer's disease already had many plaques, both neuritic and diffuse, many tangles in the cerebral cortex outside of medial lobe structures, and looking at cell loss in entorhinal cortex and the hippocampus already had substantial neuronal loss, up to 60%. These individuals, as I said, were only very mildly symptomatic, but clearly met pathologic criteria for Alzheimer's disease. In the middle are individuals who are cognitively normal at time of death, but have many neuritic and diffuse plaques, many cerebral cortical tangles, but have yet to have substantial neuronal loss. These individuals, because they still have no neuronal loss, remain normal at time of death despite a pathologic Alzheimer's disease burden. The idea here would be, if you only enroll people who are mildly symptomatic, you are trying to treat people in whom the process of synaptic and neuronal loss has already begun. It may be very difficult to demonstrate clinical benefit with such neuronal injury. The idea would be to identify people with preclinical Alzheimer's disease and treat them prior to substantial synaptic and neuronal loss. And we can identify preclinical Alzheimer's disease now using molecular biomarkers of the disease, either through CSF assays of A-beta 42 and tau and phosphorylated tau, or amyloid imaging and now amyloid PET imaging. And we identify this preclinical phase of Alzheimer's disease as lasting at least 20 years, 2 decades, maybe more, in which the individuals are cognitively normal. But as the accumulating pathophysiology builds, then additional aspects occur, and the individual have their synaptic and neuronal integrity breached, and that transitions them to the symptomatic phase of Alzheimer's disease, which is much less in length, in duration, than the preclinical phase. So we don't want to wait until people are symptomatic, even very mildly symptomatic, because they've already had substantial synaptic and neuronal injury. Additionally, once people are symptomatic with Alzheimer's disease, it's unlikely that amyloid beta monotherapies will provide substantial clinical benefit, because the symptomatic phase is accompanied by multiple co-pathologies that are not addressed by the A-beta monotherapy. It may be prudent to consider combination therapies that target not only A-beta, but the other important co-pathologies. But perhaps the most compelling rationale for therapeutic considerations is to target with anti-amyloid therapies individuals prior to symptom onset. The goal here would be to delay or even prevent Alzheimer's disease dementia. We think about the timeline using molecular biomarkers of Alzheimer's disease. The first abnormality seems to be a decrease in the amount of CSF A-beta 42 relative to CSF A-beta 40, which occurs some 25 years prior to symptomatic onset. Some 15 years prior to symptomatic onset, we begin to see elevation of CSF tau and phosphorylated tau and amyloid PET abnormalities. Most AD trials target individuals who have symptomatic Alzheimer's disease. Again, it may be too late to initiate therapies there, especially monotherapies. So there is a move to target the preclinical Alzheimer's disease, cognitively normal people who have Alzheimer pathophysiology, but have not yet experienced substantial disruption of synaptic and neuronal integrity. And there are several trials in both late onset Alzheimer's disease, A4 and A4-5, as well as in individuals with autosomal dominant Alzheimer's disease, the Alzheimer Prevention Initiative and the DIAN-TU, that are intervening with anti-amyloid and other therapies in individuals who show the amyloid and other Alzheimer pathophysiology as documented by a positive amyloid PET scan. This would be secondary prevention because these individuals already have the pathophysiology accumulating in the brain in the preclinical phase. There also now is consideration of primary prevention, just at the start or even before the start of any A-beta abnormality. These are, and there's another study, the A3 study, that is close to this primary prevention paradigm as well. These studies obviously are going to take decades to know whether they are effective in delaying or preventing the onset of symptomatic Alzheimer's disease. What cohorts might be considered for prevention trials? Autosomal dominant Alzheimer's disease has several important advantages. Mutation carriers almost all develop Alzheimer's dementia, and we know about the age that they will because it is their particular mutation and their family status gives us a good chance of knowing when they will become symptomatic. They have Alzheimer's disease pathology at least 2 decades before the symptomatic onset is expected. And because it's early onset disease, they are free from comorbid disorders that accompany age and that can contribute to dementia. We know the etiology of autosomal dominant Alzheimer's disease, overproduction of A-beta 42. A prevention trial thus will be the test of the amyloid cascade hypothesis. The disadvantage for using an ADAD cohort is that it's very rare. Sample sizes are very low. Late onset Alzheimer's disease also has advantages for prevention trials because it is very common. Matter of fact, therapeutic success in late onset Alzheimer's disease will benefit millions of people worldwide. The disadvantages of the LOAD cohort: we don't know that someone who has an amyloid PET scan that is positive but remains cognitively normal, we're not certain that they all will progress to symptomatic Alzheimer's disease, and we certainly don't know if they do when that might be. Hard to predict. Plus, these are older adults and they have many co-pathologies beyond Alzheimer's disease that will contribute to cognitive and functional decline. This is a photograph of the first known individual with Alzheimer's disease. This is Auguste Dieter, Professor Alwas Alzheimer's original Alzheimer patient. She developed symptoms at age 47. It's been speculated that she had autosomal dominant Alzheimer's disease. That's not proven, but it's certainly under consideration. So learning about the genes, amyloid precursor protein gene, presenilin 1, presenilin 2, that when mutated cause Alzheimer's disease has given us great understanding about Alzheimer pathophysiology and enables the development of transgenic mouse models to study Alzheimer's disease. We know that each offspring of an affected parent has a 50% chance of inheriting the mutation. If they do, those asymptomatic mutation carriers are destined to develop early onset symptomatic Alzheimer's disease. The Dominantly Inherited Alzheimer Network and its trials unit here at Washington University began a prevention trial in the DIAN cohort of autosomal dominant Alzheimer's disease. This was led by Randy Bateman, and subsequently, Randy has been joined by Eric McDade. This is an international adaptive trial design with 2 treatment arms, solanezumab and gantenerumab. We know from preliminary analysis that both drugs engage their target, that is, gantenerumab addresses fibrillary aggregated amyloid beta and solanezumab soluble A beta. We know that they reduced the amyloid burden, as in the cerebral amyloid burden is determined by amyloid PET scans, and gantenerumab in addition seemed to reduce the CSF total tau and p-tau. A parallel study, the Alzheimer prevention study led by Eric Ryman, Pierre Theriault from Banner Health in Phoenix, Arizona, and Francisco Lopera in Antioquia, Colombia, was launched a year after the DIAN trials unit in the largest known autosomal dominant Alzheimer's disease kindred, individuals with a presenilin mutation E280A in Colombia, South America. Here they're looking in mutation carriers at another anti-amyloid agent, crenezumab. So the overall aim of the DIAN network was to assess the sequence of abnormal Alzheimer biomarkers in asymptomatic mutation carriers in comparison with their siblings who do not have a mutation, to study pure Alzheimer's disease without age effects and without comorbid disorders. Why could such a cohort be assembled? It's very difficult because there are fewer than 1,000 known families with autosomal dominant Alzheimer's disease worldwide. And even within these families, they're often dispersed geographically. So we had to establish a global network to longitudinally assess these family members with a standard and uniform protocol. But it is an ideal cohort for prevention studies. We know when symptoms will begin in the majority of these individuals, so we can intervene with therapies years or even decades before the expected clinical onset. We know that autosomal dominant Alzheimer's disease is clearly due to overproduction of A beta. If you will, it's a pure form of Alzheimer's disease, lacking comorbid confounds. Whether these prevention trials will be successful will be very helpful in trying to understand the cause or causes of Alzheimer's disease and will guide future therapeutic development. Here is a partial example of the multiple DIAN sites that are both in the observational study and within the trials unit, and sites that are only in the trials unit, as well as some exploratory sites. You see that this now, DIAN is now on 5 continents. Here is the DIAN Trials Unit Alzheimer's Disease Prevention Trial Platform. Began with secondary prevention trial. We've mentioned this already with solanezumab and gantenerumab, because gantenerumab had the most profound effect correcting abnormal biomarkers. That secondary prevention trial is continuing with gantenerumab only in an open label extension. But a new study is being launched with lecanemab, another anti-amyloid agent sponsored by ASI, and will soon be joined by a anti-tau drug. So that will be towards the combination therapy I mentioned, treating not only amyloid, but also tau. And a primary prevention trial is being planned as well. The drug selection process is underway. In late onset Alzheimer's disease, also in 2013, Risa Sperling and Paul Asen launched the A4 study, anti-amyloid treatment in asymptomatic Alzheimer's disease, for individuals who do not come from families with a known mutation, but are older adults who are cognitively normal but have a positive amyloid PET scan. They were randomized to either the anti-amyloid antibody solunizumab or placebo. That study is closing. We're awaiting the results, but in the meantime, the AHEAD study has begun. The AHEAD 3 is cognitively normal older adults who have questionable positive amyloid PET burden. They will be treated with lecanemab, an anti-A-beta monoclonal antibody. The AHEAD A4-5 study is like the A4 study, cognitively normal older adults with a known elevation of amyloid PET scan. They also will be treated with lecanemab. Another AD prevention trial in late onset Alzheimer's disease was with pioglitazone, and it was called the Tomorrow study, but it was terminated several years ago because the drug did not delay the onset of symptomatic Alzheimer's disease. Now, can we expect results from these trials in autosomal dominant Alzheimer's disease to extrapolate to late onset Alzheimer's disease? Well, I think there's the possibility that they will. If we look at the clinical, pathologic, and biomarker presentations of autosomal dominant Alzheimer's disease, in many respects, they're identical to late onset Alzheimer's disease. Both typically begin with amnestic deficits. Of course, they have develop widespread cognitive and functional decline. Both have amyloid plaques, tangles. Both are likely to have Lewy bodies, or many of them will have Lewy bodies in addition to the Alzheimer's pathology. Late onset Alzheimer's disease, in addition to synucleinopathy, also has TDP-43, grain disease, and other co-pathologies. Both express hippocampal and whole brain atrophy. Both have elevated amyloid PET scans in the cerebral cortex. In ADAD, also some individuals have increased retention of amyloid PET in basal ganglia. FDG PET shows hypometabolism in the parietal occipital regions in both. CSF beta is decreased, CS tau is increased. Big difference in the age at onset. Midlife, 45 years for ADAD, and late life, over 70 years for LOAD. Disease duration, surprisingly, is not all that different. If a drug is shown to prevent symptoms in Alzheimer's disease, will it also work at LOAD? We tried to address this with a comparison study of the DIAN cohort with a large international cohort, the Alzheimer's Disease Neuroimaging Initiative. Both DIAN and ADNI are international programs that evaluate multimodal Alzheimer's biomarkers. DIAN was funded beginning in 2008, autosomal dominant Alzheimer's disease, mean age at onset 46 years. The ADNI cohort began in 2004. It looks at late onset sporadic Alzheimer's disease and is about 3 decades older. Because DIAN began after ADNI, we tried to adopt the ADNI imaging protocol so we could facilitate a comparison, but over time, those protocols morphed, evolved. For example, ADNI changed its amyloid tracer from PIB to florbetapir, and so they're no longer identical. Here are the individuals we had available for comparison. In DIAN, we looked at only mutation carriers, 291. In ADNI, 559 participants. In DIAN, 63% of the individuals were cognitively normal. In And in ADNI, only 13% were cognitively normal. The age difference is dramatic, about 3 to 4 decades higher, older in ADNI. This meant we could not match the cohorts on age or age at age because of this big difference. So what we decided to do was to bind them together at the time that they developed symptomatic onset, regardless of what age it was. We had these variables available for comparison. We reprocessed both the imaging and the cerebral spinal fluid assays so that they were run simultaneously on the same platforms. And we had a number of cognitive variables that were administered in common to both cohorts. Looking simply at hippocampal volume, we see in red spaghetti plots of DIAN participants and in black spaghetti plots of ANDI participants. Remember, very few ANDI participants were available prior to symptomatic onset. The majority were already symptomatic when they joined ANDI. So we have fewer individuals prior to the age at symptomatic onset, which is on the x-axis. But we do see prior to symptomatic onset in ANDI a greater reduction in hippocampal volume prior to symptomatic onset, consistent with the well-known effect that hippocampal volume loss occurs as a function of age. There is some evidence for that in the DIAN individuals, but both show a marked increase in the rate of hippocampal volume loss when Alzheimer dementia symptoms begin. And it looks that after symptomatic onset, the rate of decline is more aggressive in DIAN. We see something similar with cortical mean amyloid burden, amyloid PET, converted to centiloid units. Both DIAN and ANDI accumulate amyloid as they approach the age at symptomatic onset. After symptomatic onset, again, an inflection point, the rate of amyloid burden increases, but again, slightly more aggressive in DIAN. For CSF tau and phosphorylated tau, they also accumulate increased levels as they approach the age of symptomatic onset. Here we do not see a clear inflection point, and in many cases, both the DIAN and ANDI rates here are superimposed. So what can we say? There are clear differences between autosomal dominant Alzheimer's disease and sporadic late onset Alzheimer's disease. Load certainly is not exclusively an amyloid process. Age, ApoE effects, co-pathologies all contribute to the dementia program. Autosomal dominant Alzheimer's disease also may not be exclusively related to overproduction of amyloid. The mutations, particularly in gamma secretase, may influence other substrates that are for gamma secretase, and we don't know the effects of the mutation on those substrates. Perhaps they have deleterious effects on brain function as well. For example, Notch is a substrate for gamma secretase. The course of symptomatic Alzheimer's disease, particularly after symptomatic onset, is more aggressive than that of late onset Alzheimer's disease. But in common, both cohorts have a long asymptomatic period and nearly identical Alzheimer's disease molecular biomarker profiles. At symptomatic onset, there is often an inflection point for the biomarkers. Both groups show increasing amyloidosis, increasing tauopathy, progressive hippocampal atrophy, and I didn't show the data, but a clearly progressive cognitive decline. This gives us cautious optimism that lessons learned from autosomal dominant Alzheimer's disease will translate to late onset Alzheimer's disease. I want to list all of the DIAN and ANDI investigators that contributed to this comparison study. The manuscript reporting these results is under consideration at a journal. I wanted to also note that just this month, February 2022, we lost a major player in the Alzheimer world. John Trojanowski from the University of Pennsylvania died at age 75. So to close, I'd like to indicate that we always will continue to evaluate drugs that may be successfully treating symptomatic Alzheimer's disease. By far, the majority of people with symptomatic Alzheimer's disease need truly effective therapies. There's a huge unmet need. In this regard, we need to explore combination therapies that address multiple targets in addition to amyloid that are likely to be more successful than amyloid monotherapies. Secondary and now primary prevention strategies targeting amyloid and perhaps other pathologies may be successful in delaying or even preventing the onset of symptomatic Alzheimer's disease. With that, I thank you for your attention, and I'm happy to try to address questions. Thank you very much.
We thank Professor Morris, a wonderful talk. Because the time is limited, we only allowed 1 or 2 questions. If there are any questions, okay, I take advantage. I ask Professor Morris, in your talk, you did not mention about the blood-based biomarkers. Do you think the blood-based biomarker can play a role in identifying the preclinical AD patients?
So thank you for that question. I'm very optimistic about the promise of plasma biomarkers to serve as a much more affordable and much more accessible a way to support the diagnosis of Alzheimer's disease. We know that the correspondence of a positive plasma A beta test with amyloid PET burden is something on the order of 90%. And we're looking to combine that now with plasma, not only plasma A beta, but also plasma tau and tau phosphorylated at various sites, and also plasma NFL, neurofilament light. And I think in combination that eventually we may find that we can replace cerebral spinal fluid assays and amyloid PET and tau PET with plasma biomarkers. I'm very hopeful about that.
Okay. Any question from Professor Fu? Or Dr. Huang, Yikai? Okay. My another question is that there is some non-pharmacological intervention to prevent dementia, like a finger trial. Do you think non-pharmacological treatment can help preclinical AD patients?
So that's another very good question, and it has lifestyle interventions and modifications have a lot of positive aspects to them. I think that a healthy lifestyle does reduce the risk of Alzheimer's disease. In many developed countries, we are seeing a reduced incidence of Alzheimer's disease with control of cardiovascular risk factors and so forth. So I think that has great promise. I don't think that once the disease process begins in the brain, that we'll be able to rely upon lifestyle factors alone for treatment. So I think we need to, yes, advocate for healthy lifestyle, reduced risk, But once the disease starts, I think we're going to require pharmacological therapy.
Okay, thank you. It's good. Good question. And there is a question. Do you think that it is valuable to do preventive trial in APOE4 area for late onset Alzheimer's disease?
So I'm sorry, I may not completely understand the question, but I'm glad that it's been raised about the role of apolipoprotein E and particularly the E4 allele. I think that is certainly playing a role in how the current therapies may exert their benefit or not. But I would also like to point out that I think that ApoE4 itself could be a therapeutic target, and we haven't spent enough time considering that. So I think that's a very good point.
Okay, thank you. Because there is no other questions, so we thank Professor Morris again for his wonderful talk. And okay, sorry, there is another question. What is your opinion about p-tau 231 and p-tau 181? Difference isoform.
Another excellent question. We are finding that the standard tau phosphorylated at position 181 serves us well, but tau phosphorylated at other sites may be even more informative. Our own work at Washington University is focused on p-tau 217, but other positions are important as well. And we may find even better predictive value for tau phosphorylated at additional sites beyond 181. So good question.
Okay, thank you. Is ApoE4 considered in autosomal dominant AD?
So another very good question. ApoE4 does not seem to affect autosomal dominant Alzheimer's disease. I didn't point it out in my presentation, but the symptomatic and asymptomatic mutation carriers in the autosomal dominant families in DIAN have the same frequency of ApoE4 as their sibling non-carriers. There is no increased ApoE4 in autosomal dominant Alzheimer's disease.
Okay. Oh, yeah, we can work for 1 second. Okay. Because the time is limited, we have to close Professor Morris' talk here. And we thank Morris very much. Thank you again. Thank you again.
Thank you. I appreciate the honor. Thank you.
I hope you can visit Taipei or Taiwan again in the near future.
I would look forward to that. Thank you.
Thank you. Okay.
Bye-bye, everyone.
Bye. Goodbye. Thank you.